Because secondary metabolites and nutrients often determine the resistance or susceptibility of plants to herbivore attack, there has been great interest in discovering factors that influence the concentration of these compounds. Fritz and Sims 1992, Rosenthal and Barenbaum 1992, Oriens et al. 2003. Like other traits, variation in phytochemistry results from genetic factors, environmental sources, and interactions between the two, namely gene-environment interactions. Genetic variation among individual plants for resistance traits is a prerequisite for evolutionary change and has been central to the development of evolutionary theories on plant-insect interactions such as plant defense and coevolution, Stamp 2003, Thompson 2005. Moreover, genetic variation in plant traits has allowed plant breeders or genetic engineers to develop varieties and transgenic crops resistant to A, B, dist and Ce. Pest herbivores Borojevic 1990, Boss and Caligari 1995, Christou et al. 2006, Ferry et al. 2006. Also, environmental factors such as nutrient availability, drought stress and elevated atmospheric CO2 are all known to influence the concentrations of nutrients and chemicals in plants. These factors in turn influence plant resistance, vigor and stress. Plant conditions that have played a central role in theories predicting herbivore outbreaks. Price 1991A, Amack and Leather 2002, Hubbardie and Dino 2004. For these reasons, it is important to explore the genetic and environmental causes of phytochemical variation in plants and review the hypotheses extending from this variation that have been proposed to predict herbivore performance and population dynamics. 4.3.11 Genetic basis for plant resistance. There are many studies documenting genetic control of the concentration of both primary metabolites such as nitrogen, Caritas 1992, McPherson and Frey 1991, Zengrel and Barenbaum 2004, chemicals like alkaloids, phenolics and terpenoids Barenbaum and Zengrel 1992, Fritz and Sims 1992, Shanley and Bergelson 2000, Oriens et al. 2003, Trichomes Kenyon Barber 1992, Van Dam et al. 1999, and Plant Architecture, Rogers and Whitney 2006. In many of these studies, heritable genetic variation in phytochemistry or other plant traits influence the density of specific herbivores or the composition of the herbivory community. A good example involves the parsnip webworm Depressaria pastinacella, Lepidoptera, Oecophoridae, that feeds selectively on wild parsnip, Pastinaca sativa, a plant species that produces toxic furanocoumarins, Zengrel and Barenbaum 1990. Plant genotypes that are avoided by webworms in the field contain high concentrations of furanocoumarins, and when fed to caterpillars in the lab result in reduced performance. In contrast, field-attack genotypes have lower concentrations of furanocoumarins and foster much higher larval growth rates. In managed systems, the genetic basis of resistance has been shown by comparing the concentration of defensive metabolites or trichome characteristics between resistant and susceptible crop varieties, Kennedy and Barber 1992, Sadasivam and Tyumanov in 2003. For example, the concentration of cucurbitaceans triterpenoids in squash is under genetic control and varieties with high concentrations are resistant to attack by cucumber beetles Diabrotica. Genetic-based variation also occurs in the concentration of alkaloids in tomato and potato, and monoterpenes in pines and mints. Although there is clear evidence for heritable variation in phytochemical-based resistance, environmental factors often influence the expression or strength of resistance, suggesting a gene-environment interaction. An example of a gene-environment interaction involving chemicals is evident in wild radish, Raphanus raphanistrum, and the induction of glucosinolates by larvae of the cabbage butterfly, Pieris rapi, Agrawal et al. 2002. Genetic variation for glucosinolate concentration was not detected in undamaged plants, but was found in plants damaged by butterfly larvae. Thus, the expression of glucosinolates in radish depended on environment, namely exposure to herbivora. The breeding system of a plant and its effect on herbivora provides additional evidence for genetic-based resistance. Carr and Eubanks 2002, Hayes et al. 2004, Hull Sanders and Eubanks 2005. Inbreeding depression resulting from self-pollination often promotes detrimental changes in plants including resistance traits and tolerance to herbivora. For instance, inbred lines of Mamulus gutatus, Scriphilariaceae, were both less resistant and tolerant to attack by the spittlebug, Felinus spumarius, Hemiptera, Cercopidae. A study of the herbivores in morning glory, Ipomea hederacia convolvulaceae, found that the generalist aphid Aphis gossypii grew faster on inbred plants, whereas specialized herbivores such as the tortoise beetle, Deloyologitata coleoptera, Chrysomelody performed best on vigorous outbred lines. Variation in herbivore performance in this system was explained by the differential responses of the various herbivore guilds to plant stress and vigor between inbred and outbred lines. The study of plant hybrid zones in poplars Populus, willows, salix, and eucalyptus provides additional evidence for genetic-based resistance, and in several cases links resistance to phytochemistry. Witham 1989, Fritz et al. 1999, 2003, Dungy et al. 2000, Oriens et al. 2000, Bangert et al. 2006. It has been argued that hybrids are more susceptible to herbivore attack than the parental species because recombination breaks up codapted gene complexes that control for resistance traits. Thus, hybrids might act as herbivore sinks, where herbivores accumulate and flourish, a hypothesis that was generated by the observation that the gall-inducing aphid Pemphigus betae was far more abundant on hybrids between Fremont cottonwood, Populus fremontii, and narrowleaf cottonwood, Populus angustifolia, than on either parent. Witham 1989. Indeed, there is also evidence from eucalypts that some insect herbivores are more abundant on hybrids, and that resistance across parents and hybrids is roughly correlated with phenolic glycoside and terpenoid content, respectively. In the poplar and eucalypt systems, hybrid susceptibility cascades up to affect the composition and abundance of the herbivore assemblage and the associated arthropod community at large. Dungy et al. 2000, Wimp et al. 2005, Wimp and Witham 2007. However, there are clear exceptions to the hybrid sink hypothesis, as evidenced by the lower abundance and diversity of some leaf mining and galling herbivores oak, Quercus, and willow, Salix hybrids Boeklen and Spellenberg 1990, Fritz et al. 2003. Moreover, if codapted gene complexes controlling for resistance are disrupted during recombination, then F2 hybrids should exhibit greater susceptibility than F1 hybrids. Although this is certainly the case for some insect herbivores on hybrid willows, it is not true for others, nor is it the case for all herbivores on hybrid eucalypts. Dungy et al. 2000, Fritz et al. 2003. Exceptions to the hybrid sink hypothesis may involve the breakup of gene complexes controlling host recognition. For example, if a herbivore uses both parental species, but employs different cues such as leaf volatiles to locate each host, then recombination may disrupt host recognition and result in lower densities on hybrids. Despite the variable responses of herbivores, hybridization studies provide convincing evidence for the genetic basis of resistance. 4.3.12 Environmental influences on plant condition, resistance and herbivore dynamics. Environmental factors such as nutrient runoff and deposition, precipitation, temperature, salinization of soils, air pollution and elevated atmospheric CO2 can all influence the productivity, vigor and phytochemistry of plants with extended effects on herbivore performance and population dynamics. White 1969. With an increasing risk of global warming, changing patterns of drought and precipitation e.g., El Niño events, and escalating nutrient subsidies from agricultural and urban lands into natural systems. Boyer et al. 2002, Howarth et al. At opposite ends of the plant physiological spectrum, two hypotheses, namely the plant stress hypothesis and the plant vigor hypothesis, have figured prominently in explaining how environment-related changes in plant condition influence herbivory attack. These two hypotheses are intimately linked at the level of the individual plant because fluctuating rainfall and temperature can lead to drought stress in one season or year and vigorous plant growth at other times, exposing herbivores to a broad range of phytochemistry and plant growth dynamics. 4.3.13 Plant stress hypothesis. Historically, drought stress has been advocated as a major factor underlying outbreaks of herbivorous insects. White 1969, Matson and Hawk 1987a, b. See also Chapter 11 on population dynamics. Based on observed outbreaks of the Psyllid Cardiospina densitexta on water-stressed Eucalyptus fasciculosa trees in Australia, White 1969 formulated the plant stress hypothesis (PSH), asserting that outbreaks result from changes in plant physiology, specifically available nitrogen, during times of prolonged water deficit. When plants are water-stressed, protein metabolism and amino acid synthesis are impaired. Xiao 1973. Under such conditions, proteins can be hydrolyzed, resulting in increased levels of free amino acids. Broadbeck and Strong 1987. Also, to offset the low osmotic pressure resulting from water stress, many plants produce nitrogen-containing osmoprotectants, Aspinall and Peleg 1981. Because nitrogen is so limiting for insect herbivores, White 1993 suggested that stress-induced increases in available plant nitrogen could result in the improved growth and reproduction of phytophagous insects and thus promote population outbreaks. However, since the inception of the PSH, an increasing number of experimental studies have failed to support its predictions. Larson 1989, Mopper and Witham 1992, Waring and Cobb 1992, Khorosheva et al. 1998a, Hubbardy and Dino 2004. Although some phytophagous insects benefit on water-stressed plants, many others do not and are very adversely affected. Such mixed support for the PSH led to modified predictions based on a greater understanding of the physiological responses of plants to water deficit and the effects on specific feeding guilds. Larson 1989. For example, besides increases in nutritional compounds during water stress, turgor pressure and water content decrease. Xiao 1973, Inba et al. 2001. Alala chemicals often elevate. Gershenson 1984, Matson and Hawk 1987a, b. Inba et al. 2001, and plant growth is reduced. Price and Clancy 1986, Price 19918, thus compromising the potential benefits of elevated nitrogen to insect herbivores. Also, certain feeding guilds were thought to respond more positively to water-stressed plants than others because they differentially experience changes in plant nutrition, alala chemistry and growth. Specifically, phloem and cambium feeders, because they feed in vascular tissues with low concentrations of alala chemicals, were suggested to respond more positively to plant stress than free-living chewing insects and gall formers. Larson 1989. Despite such refinements, however, recent assessments have failed to find consistent support for PSH and do not explain the discrepancy between early field observations and recent experimental studies, reviewed in Hubbardy and Dino 2004. A recent meta-analysis of published experimental studies challenges the historical view that herbivorous insects exhibit elevated performance and outbreak dynamics on water-stressed plants. Hubbardy and Dino 2004. Overall, strong negative effects of water stress were found on the performance of sap-feeding insects, e.g., aphids and leafhoppers, and gall-inducers, e.g., sawflies and cystidiomyiid flies, and inconsistent responses resulted for free-living chewers and leaf miners, e.g., lepidopterans and beetles. Only borers, bark beetles and longhorn beetles showed consistent positive responses on drought-stressed hosts. The positive response of bark beetles likely results from reduced tree resistance during attack, because oleoresin exudation pressure is lower under conditions of drought stress. Matson and Hawk 1987a, B. Lorio et al. 1995. To partly explain the discrepancy between the observed outbreaks of sap feeders, e.g., phloem-feeding psyllids, on water-stressed plants in nature and the negative effects detected in many experimental studies where plants are continuously stressed, Hubbard and Dino 2004 proposed a pulsed stress hypothesis, whereby bouts of stress and the recovery of turgor allow sap feeders to access and thus benefit from stress-induced increases in plant nitrogen. The finding that phloem-feeding aphids respond positively on intermittently stressed plants, but exhibit poor performance on continuously stressed plants, is consistent with this hypothesis. Together with knowledge of feeding guild, the phenology and intensity of water stress as they mediate nitrogen availability and plant defense likely hold the key to understanding how water stress affects the population dynamics of insect herbivores. Overall, recent assessments suggest that the great majority of insect herbivores are adversely affected on water-stressed hosts. However, White 2009 has stressed that the plant stress hypothesis should be limited to explaining outbreaks of insects feeding on senescing plant parts, senescence feeders which are responding to physiological changes in the host plant. 4.3.14 Plant vigor hypothesis. If plant stress often negatively affects many insect herbivores, then one might expect them to perform well on vigorously growing plants and plant tissues. This expectation has been formalized as the plant vigor hypothesis, which was spawned from observations on gall-inducing insects that require rapidly proliferating plant tissue for successful gall formation. Price 1991a. See also Chapter 11. For instance, gall-inducing sawflies Hymenoptera, Tenthredinidae, and wasps Hymenoptera, Cynipidae selectively attack fast-growing shoots over slow-growing ones, and they prefer vigorous-growing plants over slow-growing plants. Also, larval survival within galls can be vastly higher on rapidly growing shoots and plants, a pattern observed for several willow-inhabiting sawflies, Ura spp. And rose-attacking gall wasps, Diplolepis spp, a pattern that is not driven by natural enemies. Craig et al. 1986, Price and Clancy 1986, Price et al. 1987a, B. Roininen et al. 1988. Because tissue growth is dramatically reduced when plants experience water deficit, gall formers often perform very poorly in water-stressed plants. Besides gall inducers, other insects too show enhanced performance and density in vigorously growing plants or plant tissues. Furness and Carolyn 1977, Price et al. 1990, Price 1991a. Foresters have recognized for years that shoot-boring herbivores such as tip moths, e.g. Rhiasionia frustrana, and weevils, e.g. Hylobius palisand pisode strobi, are severe pests on vigorously growing pine seedlings and young trees, and they are far less abundant on mature trees. Also, many-insect herbivores selectively attack the new growth or regrowth of trees and shrubs that occurs after rains, artificial defoliation or mammalian browsing. Messina et al. 1993, Martinez and Wool 2003, Agrawal and Spiller 2004. Aphids, for example, achieve dense populations in the new shoots of grasses, shrubs and trees that appear following defoliation or pruning. Johnson and Leone 1991, Brown and Welker 1992. Also, moose-browsed birch trees with extensive airgrowth are more heavily attacked than non-browsed trees. Insect herbivores in a diversity of eating guilds, including sap feeders, gall inducers and leaf miners. Donnell and Hustonell 1985. However, shoot vigor is often confounded by enhanced nutrition and reduced defense. A manipulative field experiment, in which water and nitrogen treatments were applied to willow plants, allowed for an assessment of the relative contributions of shoot length, leaf protein and phenolic content to the density and survival of the gall-inducing sawfly Urolaciolepis. Waring and Price 1988. Across the array of treatment plants, shoot length was a far better predictor of sawfly attack and survivorship than were protein or phenolic content. Similarly, an irrigation experiment in grapevines, Vitis vinifera, provides convincing support for the plant vigor hypothesis without any obvious confounding effects of elevated tissue nitrogen. Don and Williams 2003. Irrigating grapevines with increasing amounts of water resulted in commensurate increases in shoot length, vegetative growth and midday leaf water potential, but not leaf nitrogen content, which in fact declined. Densities of the variegated leafhopper, Erythronera variabilis, Hemiptera, Cicadelidae, were tightly correlated with vine growth and the amount of water applied, but not leaf nitrogen. It may be though that the elevated leaf water potential on vigorously growing vines allowed leafhoppers to ingest phloem sap more easily as a consequence of elevated cell turgor. Hubbard and Dino 2004. For many herbivores, especially free-living ones that do not rely directly on tissue proliferation, isolating the effects of plant and tissue vigor from other correlated variables on herbivore performance remains a challenge. 4.3.15 Other environmental stressors affect plant resistance to insect herbivores. Abiotic stressors including salinity, pollutants such as sulfur and nitrogen dioxide, and elevated levels of atmospheric CO2 and ozone are growing environmental threats to natural systems. Rogers and McCarty 2000, Hunter 2001a, Whitaker 2001, Hamilton et al. 2005. These multiple stressors influence plant-insect interactions largely through their effects on phytochemistry, which in turn influences plant resistance and susceptibility to herbivory attack. In the coming paragraphs we will focus on several of the more important of abiotic stressors and their plant-mediated effects on herbivores. Soil salinization coupled with rising temperatures can lead to plant stress with consequences for herbivores. Rogers and McCarty 2000, Scheil and Mopper 2006. Salt stress elicits similar physiological responses in plants as does drought stress. Hasegawa et al. 2000, Manser 2000, Mopper et al. 2004. In general, salt stress promotes increases in nitrogen-containing osmoprotectants and reductions in cell turgor, water content, plant growth and productivity. Such changes should have negative consequences for herbivores, especially those that rely on high tissue water content and turgor to feed efficiently and assimilate nitrogen. Slansky and Schreiber 1985, Hubbard and No 2004. Most evidence suggests that the performance of insect herbivores is adversely affected on salt-stressed plants, especially on glycophytic plants that are not salt-adapted. A manipulative experiment in which the leaf-mining agromyzid Flycerodontha iridifora was raised on glycophytic iris plants, Iris hexagona, grown under saline and freshwater conditions showed that salinity was detrimental to the performance and survival of both the leaf miner and the iris. Shailen Mopper 2006. Likewise, on glycophytic goldenrods, Solidago, salt stress also results in the reduced performance of dipteran leaf miners and gall inducers. Martel 1998, Moon and Stilling 2002b. Not surprisingly, results there more mixed when herbivores are raised in adapted halophytic plants. Nonetheless, there is still evidence that stem-boring beetles and lepidopterans, as well as gall inducers, incur reduced performance when their halophytic host plants are salt-stressed. Hemingwand van Solen 1988, 1992, Moon and Stilling 2002a, b. Interestingly, all of these herbivores are reconcealed feeders, which often incur fitness penalties when plant growth is reduced. In contrast, salinity had no effect on the performance of lepidopteran leaf miners on C. Astor, Hemminga and van Solen 1992, and it was positively associated with the abundance of dipteran gall formers and the Planthopper Piscinotus quadricustellatus on their salt-stressed host plants. Gon Cabs Album et al. 2001, Moon and Stilling 2002a. The continuous rise in the atmospheric concentration of greenhouse gases, County 2 and ozone, since the mid-1800s has prompted ecologists and entomologists to study their effects on plant-insect enemy interactions. Lindroth et al. 1995, Hunter 2001c, Whitaker 2001, Percy et al. 2002, Monder et al. 2004. In general, when plants are grown under elevated CO2, photosynthesis, growth and biomass all increase. The accumulation of biomass dilutes the concentration of nitrogen in plant tissues by 15-25% and results in a higher C/N ratio. Also, when plants experience elevated CO2, carbon-based defenses such as condensed tannins and phenolic glycosides often increase, but there are exceptions. In general, on a poor quality diet of reduced nitrogen concentration, elevated carbon-based defenses and the diluted effects of nitrogen-based defense, it is logical to think that insect herbivores will show reduced performance and survival. However, there is considerable variation in how insect herbivores respond when fed plants grown under elevated CO2. Hunter 2001c, Whitaker 2001. A common response of many herbivores raised on plants exposed to elevated CO2 is for them to increase their feeding rate, but most herbivores cannot compensate fully and suffer some fitness penalties. As an example, when larvae of the buckeye butterfly, Junonia senia, are fed plants of Plantigo lanceolata grown under elevated CO2, they incur higher mortality rates and grow much slower. Pahur et al. 1989, 1991. Higher rates of herbivore mortality in some cases have been attributed to nutritional deficiency that results from reduced foliar nitrogen content under elevated CO2 conditions. Brooks and Whitaker 1999, Stilling et al. 2003a. Reductions in gypsy moth performance on aspen trees are associated with CO2 promoted increases in phenolic glycosides. McDonald et al. 1999. Some herbivores, in fact, appear to benefit under elevated CO2. Larvae of the lycaenid butterfly Polyamatus icarus increase their consumption rate and grow faster when reared on Lotus corniculatus grown under elevated CO2, a legume with nitrogen-based defensive metabolites. Gubbard et al. 1999. Larvae apparently perform better because concentrations of cyanogenic glycosides decrease. Similarly, populations of several aphid species increase when reared on hosts exposed to elevated CO2. Amack et al. 1997. Bessemer and Jones 1998. Whitaker 2001. Given the published data at hand, chewing insects in general appear to suffer more from elevated CO2 effects than do sap feeders, perhaps because sap feeders circumvent compartmentalized carbon-based defenses, are pre-adapted for excreting excess carbohydrate and often have symbiotes that can augment dietary nitrogen. Elevated CO2 can also affect interactions between herbivores and their natural enemies. Because feeding compensation with protracted growth is often a consequence of elevated CO2, herbivores may be at a higher risk of attack from natural enemies. Stilling et al. 1999. Lepidopteran leaf miners on several oak species, for instance, experience growth delays and higher attack rates from parasitoids and predators, mortality factors that lead to a reduced density of leaf miners on trees grown under elevated CO2. Also, coccinellid beetle larvae, Lays axarides, increase at their consumption rate of cotton aphids, Aphis gossypi, under elevated CO2, perhaps because the protein content of aphids is lower and beetles compensate with increased predation. Chen et al. 2005. The result of increased consumption by beetles is enhanced biological control of aphids. Overall, responses of herbivores to elevated CO2 are complex and dependent herbivore identity, plant species and genotype, soil nitrogen availability and interactions with natural enemies. Hunter 2001a, Whitaker 2001, Stilling et al. 1999, 2003a, Saxon et al. 2004. Nonetheless, the consensus is that increasing levels of atmospheric CO2 will adversely affect many insect herbivores via changes in carbon-based phytochemistry. Percy et al. 2002. Ozone O3 effects on phytochemistry and insect herbivores appear to be less than those of elevated CO2. Holopainen and Co. Diaresis SSI 1998, Costa et al. 2001, Whitaker 2001. Elevated O3 had no effect on the concentration of amino acids, sugars, monoterpenes, ricin acids and total phenolics in Scots pine, Pinus sylvestris, and there were no negative effects on the reproductive rate of the gray pine aphid, Schizolochnus spinetti, Kinolainen et al. 1994. However, in trembling aspen Populus tremuloides, elevated ozone-reduced tree growth, lowered concentrations of phenolic glycosides, but increased cuticular waxes, factors that were associated with improved performance and a greater potential for population outbreaks in the forest tent caterpillar, Mollicosoma dystria, Percy et al. 2002. It is noteworthy that in this aspen system there are significant interactive effects between elevated CO2 and O3, such that one gas can offset or exacerbate the effects of the other on phytochemistry and herbivores. For instance, elevated CO2 reduced the positive effect of O3 on tent caterpillar performance and on the density of the aphid Chaetophorus stevensis. In this system, the long-term suppression of herbivore populations by natural enemies is expected to be lessened under conditions of elevated CO2, O3 or both. Thus, changes in atmospheric gases and their plant and enemy-mediated effects in herbivores appear to differ between systems. Percy et al. 2002, Stilling et al. 1999, 2003a. Atmospheric pollutants such as sulfur dioxide, SO2, and nitrogen monoxide and dioxide, NO and NO2, also influence herbivores via altered plant phytochemistry. Whitaker 2001, Jones and Payne 2006. Plants downwind from pollution sources and those growing close to busy roads tend to support higher densities of herbivores. For instance, pine budmuds, Exotelea dodicella, and spruce needle miners, Epinotia tudela, achieve much higher densities on conifer trees growing in close proximity to pulp mills, a major source of SO2. Oxinen et al. 1996. Most experimental studies show that insect herbivores increase in response to elevated SO2 and NO2, Heliodiaresis vara and Va diaresis Isa diaresis Nen 1993. One mechanism by which SO2 and NO2 increase the susceptibility of plants to herbivore attack appears to be altered nitrogen metabolism, Deepak and Agrawal 2001, Whitaker 2001, Morikawa et al. 2004. Also, both SO2 and NO2 can reduce the searching behavior of parasitoids, which leads to lower rates of host parasitism, Gate et al. 1995. There are also important interactive effects of air pollutants, such that the combined effects of elevated SO2 and NO2 on herbivore performance, e.g., Elatobium obitinum, are greater than the singular effect of each dioxide alone. Altogether, alarming changes in pollutants and greenhouse gases with expected, but complex, effects on phytochemistry should foster even more aggressive research in this critical area of environment-mediated plant-insect interactions. 4.4 Plant Defense Hypotheses. The coevolutionary, arms race, between plants and herbivores emphasizes an ongoing reciprocal interplay whereby each player exerts reciprocal selective pressure on the other resulting in evolutionary change, Ehrlich and Rabin 1964. So far, we have taken a very herbivore-centered approach to plant-insect interactions by examining the consequences of plant defenses for phytophagous insects and the counteradaptations which allow herbivores to cope. We will now take the plant's perspective and explore the distribution of phytochemicals within and among plants and the many factors that bear on their expression. Because plants grow, compete with other plants and reproduce, they can't allocate all available energy to defense. Moreover, plants exist in a great diversity of habitats that vary in resource availability, e.g., nitrogen, water and light, and resource limitation is known to affect patterns of allocation to defense in plants. Also, there is tremendous temporal and spatial variation in herbivore and pathogen pressure, within and among plants and plant populations, thus affecting natural selection for defense. In the context of the diverse selective pressures that bear on plants, several hypotheses have been put forth to explain the existing variation in plant defenses, reviewed in Stamp 2003. In a nutshell, the optimal defense hypothesis attempts to explain variation in the allocation of plant defenses in relation to the plant's risk of attack, the value of the attacked tissue to the plant, and production costs of defenses. McKee 1974, 1979, Feeney 1975, 1976, Rhodes and Cates 1976, Rhodes 1979. Mixed results and theoretical advances prompted the development of additional hypotheses including the carbon-nutrient balance hypothesis, Bryant et al. 1983, the growth rate hypothesis, Coley et al. 1985, and the growth differentiation balance hypothesis, Herms and Matson 1992. Although controversial, these hypotheses establish a framework for exploring patterns of plant defense and set the stage for the development of improv theory. Thus, we will examine each of these hypotheses in the forthcoming sections. 4.4.1 Optimal Defense Hypothesis. The optimal defense hypothesis predicts the allocation of plant defenses in relation to the plant's risk of attack, the value of a tissue to the plant and the cost of the defense. McKee 1974, 1979, Feeney 1975, 1976, Rhodes and Cates 1976, Rhodes 1979. The optimal defense hypothesis assumes that plants evolve and allocate defenses in a way that maximizes individual fitness. Thus, plants and plant parts should evolve defenses in direct proportion to their risk from herbivores. Historically, the plant apparency hypothesis, within a plant, tissues should be defended in proportion to their value to plant fitness. Allocation to defense varies depending on herbivore presence or absence. Allocation to constitutive versus inducible defenses, and defenses are costly because they divert resources from other essential plant needs, such as growth and reproduction. Stamp 2003. Overall, the optimal defense hypothesis predicts that plants should allocate more to defense when the benefits outweigh the costs. The predictions of the optimal defense hypothesis further presume the following three conditions. 1. Defenses reduce herbivora. 2. There is genetic variation in secondary metabolites on which selection can act. 3. Herbivora is a major selective force underlying The production of secondary metabolites. In general, there is support for these presumptions. First, there is an enormous literature documenting that secondary metabolites and other plant defenses indeed reduce levels of herbivora, Rosenthal and Berenbaum 1991, 1992, Carbon and Baldwin 1997. Second, there is widespread evidence that the concentration of allylochemicals such as alkaloids, glucosinolates and furanocoumarins is under genetic control, Zengel and Berenbaum 1990, Van Dam and Vreeling 1994, Mauricio 1998, Shanley and Bergelson 2000, Oriens et al. 2003. Third, there is building evidence from experimental studies that insect herbivores can act as a major selective force underlying the evolution of plants' secondary metabolites, Sims and Roscher 1989, Mauricio and Roscher 1997, Shanley and Bergelson 2000. For instance, by manipulating the abundance of insect herbivores on Thessalionaceous plant Datura stramonium, it was shown that insects acted as selective agents on the concentration of 2-tropane alkaloids, even when levels of herbivora were relatively low, Shanley and Bergelson 2000. There are also correlative studies showing that on a geographic scale concentrations of secondary chemicals or resistance factors in plants are positively associated with herbivore abundance, a pattern consistent with the view that herbivores are the agents of selection, Hay 1991, Pennings et al. 2001, Stamp 2003. An eye-catching example occurs in the Bothnian archipelago where meadowsweet, Filipendula rosaceae, and the specialist leaf beetle Galaruselatinella coleoptera, chrysomelody colonize islands that are subject to postglacial rebound, Stenberg et al. 2006. Postglacial rebound is the rise of land masses that were depressed by the enormous weight of ice sheets during the last glacial age. The result is an array of islands that range in age from very recent to over 1,000 years, creating the opportunity to examine plant resistance traits in meadowsweet that have had very discrepant historic exposure to herbivores. Meadowsweet in old islands, those with long-standing exposure to herbivores, had significantly higher concentrations of condensed tannins and individual phenolics than plants growing on youngest lands. Moreover, beetles performed poorly on well-defended plants from older islands and developed rapidly on less defended plants in younger populations. These results are entirely consistent with the view that herbivores play a selective role in the evolution of phytochemical defense. Indirect evidence that herbivores are the active agents of selection for secondary metabolites comes from plant lineages that have lost a particular allylochemical when they evolve a substitute defense, Futuma and Keyes 1992. For example, Central American acacia trees that harbor predaceous ants have lost the cyanogenic glycosides that occur in non-ant defended relatives, Rare et al. 1973. Other studies, however, suggest that herbivorous insects are not the major selective force underlying the abundance and diversity of allylochemicals, as has been suggested fourth occurrence of tannins in North American trees, Iris et al. 1997. The opportunity to test the selective effects of herbivora on allylochemistry arises when plants have been introduced to novel habitats in the absence of their native herbivores, Daler and Strong 1997, Seaman and Rogers 2001, Castells et al. 2005, Lewis et al. 2006. By comparing herbivora and phytochemistry on invasive plants in their native and novel range, one can test the expectation that, if costly, plants should evolve reduced defense where herbivore pressure is low. Introduced populations of the Chinacetalo tree, Sapium seabiferum, in the southern United States, where herbivora is rare, contain undetectable amounts of tannins, a pattern that contrasts with the high concentrations of tannins in its native China, where herbivora is prevalent, Seaman Androgers 2001. For invasive plants in general, however, evidence is mixed, with examples in which allylochemistry is not reduced in the invaded area despite a low level of herbivora compared to that in the native range, Willis et al. 1999, Lewis et al. 2006. Other variables, such as plant pathogens, resource availability and abiotic factors, e.g., UV light, can complicate patterns of plant defense, making it difficult to isolate herbivora as the sole force underlying the occurrence of allylochemicals, Bostock 2005, Carroll and Berenbaum 2006. A major prediction of the optimal defense hypothesis is that there is a cost to defense and the materials and energy allocated to defense cannot be allocated simultaneously to other functions like reproduction and growth, Stamp 2003. Accordingly, when herbivores are absent, less well-defended plants should have higher fitness than better defended individuals. In fact, many studies indicate that there are costs associated with the biosynthesis, maintenance and induction of allylochemicals, costs that are leveled against growth and reproduction, Baldwin 1998, Vreeling and Van Beek 1994, Bergelson and Purrington 1996, Strauss et al. 2002. Overall, there is evidence that constitutive defenses, compartmentalized allylochemicals and induced defenses are costly to the plant. For constitutive defenses in the tropical tree Cecropia peltata, tannin-rich trees incur less herbivora, but there is a trade-off between defense and growth, as evidenced by an inverse relationship between tannin concentration and leaf production among individual trees, Coley 1986. Regarding compartmentalized allylochemicals, a trade-off between defense and reproduction occurs across genotypes of Brassica, whereby an increase in myrosinase, acts on glucosinolates to form toxic isothiocyanates, was correlated with increased resistance to flea beetles Philotreta cruciferae, but at the cost of lower seed production, Mitchell-Olds et al. 1996. Isolating the cost-OPA-specific resistance factor is now possible with genetic engineering. As an example, transplanting a resistant gene into Arabidopsis thaliana reduced seed production by 34%, demonstrating a clear cost to resistance, Bergelson et al. 1996. Because there are explicit fitness penalties associated with the production of constitutive and compartmentalized allochemicals, activated allochemicals, that are not induced until herbivores attack are thought to minimize such costs. Strauss et al. 2002, Kessler and Baldwin 2004, Bostock 2005, Walsh et al. 2005. A field study with Nicotiana attenuata, a species in which the alkaloid nicotine is inducible by wound hormone jasmonic acid, demonstrates this expectation. Baldwin 1998. Induced plants were attacked less often by herbivores and survived to produce more seed than did their non-induced counterparts. However, if plants were not attacked, jasmonate-induced plants produced less seed than did non-induced plants. In this system, jasmonate-induced responses function as defenses, but are costly, and inducibility allows this species to forego these costs when defenses are unnecessary. Evaluating trade-offs between defense, growth and reproduction is complicated by the existence of a third-party trade-off with tolerance. In fact, plants cope with herbivore attack in two ways. So far, we have focused on resistance, traits that reduce damage, but plants also exhibit tolerance, traits that reduce the impact of damage on plant fitness. Stowe et al. 2000, Stamp 2003. Tolerance to tissue damage, essentially regrowth ability, involves a variety of factors, including growth rate, storage capacity, flexible photosynthetic rate and nutrient uptake, and developmental plasticity. Rosenthal and Cotan in 1994. Tolerance has likely evolved in response to herbivory, and it is under genetic control. Feinblum and Roscher 1995, Mauricio et al. 1997. Plants appear to have one of three strategies: well-developed defense and poor tolerance, well-developed tolerance and poor defense, or an intermediate. Aboth, van der Meiden et al. 1988. Thus, there appears to be a negative association between tolerance and resistance, but both can occur together in the Sami plant. Feinblum and Roscher 1995, Mauricio et al. 1997. Overall, there is clear evidence for costs of resistance to plant fitness, but detecting them can be complicated by trade-offs with tolerance, differences in production and maintenance, and induction costs among metabolites, and differences in environmental conditions. Risk of herbivore attack is another factor thought to influence the distribution and abundance of allochemicals in plants and plant tissues. Originally proposed as the plant apparency hypothesis, Feeney 1975, 1976, Rhodes and Cates 1976, risk of attack is now subsumed as a feature of optimal defense theory, Rhodes 1979. The plant apparency hypothesis posits that plants easily found by herbivores, apparent plants, should invest heavily in costly quantitative defenses such as tannins, which occur in high concentrations and are broadly effective against a diversity of herbivores including both specialists and generalists. Apparent plants include long-lived trees, shrubs and perennial grasses. In contrast, unapparent plants, due to their rarity, patchiness or short generation times, are expected to escape most herbivora, and thus selection should favor low investments in cheap qualitative toxins that are effective against all but specialized herbivores. Unapparent plants include fast-growing annuals, early successional plants and rare plants. Although there are rough correlations between plant apparency and expected allocations to defense, e.g., tannins predominate in apparent oak trees, which lack glucosinolates, whereas the opposite pattern prevails in ephemeral mustards, there are notable exceptions to this hypothesis. Stamp 2003. For example, in their defensive arsenal, some unapparent plants employ quantitative defenses, whereas even very apparent plants often contain toxins. Further complicating the predictions of the plant apparency hypothesis is the fact that quantitative allochemicals, such as some tannins and terpenoids, can have both digestibility reducing and toxic properties. Ayres et al. 1997, Langenheim 2003, Keeling and Bowman 2006. Mostprobomatic is trying to quantify the apparency of a plant to a herbivore, given the tremendous variation among herbivorous insects in their dispersal, sensitivity to plant cues and thus their ability to colonize their host plant. Feeney 1991a, Bernays Chapman 1994. In short, many herbivores locate their host plant very effectively and synchronize their life history with it, despite its rarity and fleeting existence. Given that most plant species are reattacked by a dynamically changing assemblage of many herbivore species with a diversity of host locating abilities, how does one quantify the mean apparency of a plant, the measure on which selection must act? Overall, the plant apparency hypothesis proved a useful framework when it was proposed, and contributed significantly to our knowledge of plant defense, but difficulty with measuring apparency, and the mixed empirical support remain as detractors. Stamp 2003. However, when we consider what humans eat, the preponderance of our vegetable intake is from groups of plants with originally inapparent distributions and toxic defenses, such as in the cabbage, carrot and lettuce families of plants. We do not eat tree leaves, and restrict our diet from trees to nuts and fruits. So from this broad perspective we can see that the plant apparency hypothesis accounts for the human diet effectively. This hypothesis has proved to be central in the conceptual development of plant defense theory and has general predictive power for the dichotomy in defensive syndromes between short-lived forbs and long-lived trees. However, uncertainty arises because plant apparency is difficult to assess, the predictions of risk and value are often confounded, costs are evaluated without knowledge of the tolerance-resistance relationship in plants, and environmental conditions or resource availability and stress were not originally considered. This ambiguity has led to the development of other explanatory hypotheses of plant defense and allochemical allocation. 4.4.2 Carbon-nutrient balance hypothesis. The carbon-nutrient balance hypothesis seeks to explain how the supply of carbon and nutrients in the environment affect the phenotypic expression of defenses in plants and thus resistance to herbivore attack. Bryant et al. 1983, Twomey et al. 1988, 1991, Figure 4.12. The carbon-nutrient balance hypothesis was developed originally to explain the effect of soil nutrients and shade on phytochemical defenses that extend from an altered carbon-nutrient ratio in the plant. The hypothesis suggests that when plants low nutrients, low carbon, low nutrient availability, normal light and CO2 availability, nutrient absorption, growth, opening parenthesis dash closing parenthesis, opening parenthesis dash closing parenthesis, carbohydrate concentration, C-based defense, photosynthesis, N-based defense, nutrient concentration, normal nutrient availability, photosynthesis, low light, CO2 availability, nutrient absorption, growth, opening parenthesis dash closing parenthesis, nutrient concentration, carbohydrate concentration, N-based defense, C-based defense. Figure 4.12 carbon-nutrient balance hypothesis is proposed by Bryant et al. 1983. The hypothesis was developed originally to explain the effect of soil nutrients and shade on phytochemical defenses, N-based and C-based, that extend from an altered carbon-nutrient ratio in the plant. Vertical arrows in boxes indicate an increase, up pointing arrow, or decrease, hash, in a variable. Arrows between boxes, or, show a positive effect unless a negative symbol, is indicated. The thickness of arrows indicates the relative strength of the effect. From Bryant et al. 1983. Reprinted with permission from Blackwell Publishing. Acquire resources in excess of growth demands, that these resources are shunted into the production of secondary metabolites, Lerdow and Coley 2002. Specifically, plants growing in nutrient-poor soils should allocate more resources to carbon-based defenses, e.g., phenolics, whereas those growing in low-carbon shady environments should allocate more to nitrogen-based defenses, e.g., alkaloids. The carbon-nutrient balance hypothesis further posits that nitrogen fertilization will shift defenses from carbon-based to nitrogen-based. The hypothesis is appealing in its simplicity and because it offers testable predictions. Khorosheva et al. 1998b, Hamilton et al. 2001, Stamp 2003. However, in the many tests of the carbon-nutrient balance hypothesis, results supporting its predictions are mixed. Khorosheva et al. 1998b. The domain of the hypothesis is at higher categorical levels, effectively predicting the total amount of carbon that can be allocated to carbon-based defense. However, predicting altered concentration of specific compounds requires knowledge of biosynthetic pathways. Khorosheva et al. 1998b, Lerdow and Coley 2002. For example, this hypothesis successfully predicts changes in the concentration of products of the shikimic acid pathway. As predicted, studies across a large range of plant species growing in many habitats found an increase in condensed tannins with increases in light or decreases in nutrients. Khorosheva et al. 1998b. Also consistent with this hypothesis is the phytochemical response of silver birch, Betula pendula, to fertilization. Mutikainen et al. 2000. Fertilization resulted in a decrease in the concentration of condensed tannins, which was associated with decreased tree resistance to the moth Aperita autumnata. Similarly, increases in phenolics and condensed tannins have been found under conditions of elevated CO2, which increase the C/N ratio in plants. Lindroth 1996, Lerdow and Coley 2002, Percy et al. 2002. Results are less predictable for terpenes and alkaloids, the TER products of non-shikimic acid or multiple metabolic pathways. Khorosheva et al. 1998b. Moreover, the predictability of the carbon-nutrient balance hypothesis also depends on the phenotypic plasticity of the plant. See Stamp 2003. Research emerging from tests of the carbon-nutrient balance hypothesis suggests that the defensive responses of plants to variable resources are more complicated than was originally thought. Lerdow and Coley 2002, Stamp 2003. A greater knowledge of the plasticity of plant defense in relation to environmental resources and an understanding of the biosynthetic pathways involved in the synthesis of particular defensive metabolites is necessary to refine predictions stemming from the carbon-nutrient balance hypothesis. Nonetheless, this hypothesis called attention to how resources influence constitutive and induced defenses and contributed to the emergence of the following hypothesis regarding plant defense. 4.4.3 Growth Rate Hypothesis. The growth rate hypothesis, also referred to as the resource availability hypothesis, proposes that the pattern of plant defense is largely determined by the inherent growth rate of a plant, which is ultimately determined by available resources. Coley et al. 1985. Thus, among plant species, resources limit plant growth and the optimal level of investment in constitutive defense increases with a decrease in plant growth rate. Figure 4.13. The optimal level of defense refers to the expression of defenses at maximal growth rate under conditions of optimal nutrition for each plant species. The growth rate hypothesis predicts that in high-resource environments, e.g., agroecosystems and old field habitats, competition favors fast-growing plant species with little allocation to defense because any herbivore-inflicted damage can be compensated for by rapid growth. Conversely, in low-resource environments, e.g., drier shaded habitats, resource limitations elects for slow-growing plant species with much greater allocations to defense. Such slow-growing species cannot easily compensate for tissue loss because of their slow growth, so replacement of resources lost to herbivores is more costly for plants in low-resource environments, and thus their higher allocation to defense. As predicted, there is a negative correlation between the growth rate of neotropical tree species and their tannin concentration. Coley 1987. More recently, a reciprocal transplant study in Amazonian forests involving tree species growing on nutrient-rich clay soils and nutrient-poor sandy soils found strong support for the growth rate hypothesis. Fine et al. 2004. Tree species originating from nutrient-poor soils express higher levels of constitutive defenses, tannins, than those growing on nutrient-rich clay. Overall, some studies provide support for the growth rate hypothesis, Jing and Coley 1990, Herms and Matson 1992, whereas others do not, Cates 1996. The major contribution of the growth rate hypothesis is that it makes a compelling case for the evolutionary history of resource availability on the intrinsic growth rate of plants, which in turn is the primary factor responsible for shaping the defensive profile of plants. 4.4.4 Growth Differentiation Balance Hypothesis. The growth differentiation balance hypothesis predicts how plants balance allocation between differentiation-related processes, such as defense, and growth-related processes across a gradient of environmental conditions, Loomis 1953, Herms and Matson 1992. Growth is the production of roots, stems and leaves or any process that requires substantial cell division and elongation. Differentiation is everything else, and refers to the enhancement of the structure or function of existing cells, e.g., maturation and specialization. Some differentiation traits bear directly on plant defense. For example, the production of secondary metabolites, trichomes and thick leaf cuticle are examples of differentiation-related processes, Herms and Matson 1992. Allocation to differentiation includes both the process and the products, so it subsumes enzyme and metabolite costs as well as their transport storage costs. The growth differentiation balance hypothesis of plant defense is premised on a physiological trade-off between growth and differentiation processes, Herms and Matson 1992. The trade-off occurs because secondary metabolism and associated structures, e.g., trichomes, latex canals and resin ducts, are high maximal growth rate, low maximal growth rate. 0.51.0 Defense investment, G defense per gram leaf. REALEZED. GROWTH. RE. Figure 4.13 Growth rate hypothesis is proposed by Coley et al. 1985. The hypothesis posits that the pattern of plant defense is largely determined by the inherent growth rate of a plant, which is ultimately determined by available resources. The graph shows the effect of defense investment on realized plant growth. Each curve represents a plant species with a different maximum inherent growth rate. Levels of defense that maximize realized growth are indicated by an arrow. From Coley, P.D., J.P. Bryant, and F.S. Chapin. 1985. Resource availability and plant anti-herbivore defense. Science 230, 895-899. Reprinted with permission from AAAS. Physiologically constrained in dividing and growing cells, and because they divert resources from the production of new leaves. Thus, these processes compete directly for photosynthesis, and plants face the dilemma of growing fast enough to compete with other plants while at the same time maintaining sufficient defenses to survive attack by herbivores and pathogens. The growth differentiation balance hypothesis posits that any environmental factor slowing growth more than it slows photosynthesis increases the pool of resources available for allocation to differentiation-related products such as defensive metabolites, Herms and Matson 1992, Glynn et al. 2003. For instance, growth is reduced substantially by shortages of nutrients and water, whereas photosynthesis is less sensitive to such limitations. In such instances, carbohydrates accumulate in excessive growth demands and thus can be converted to secondary metabolites without much cost to plant fitness. Accordingly, the growth differentiation balance hypothesis explains how the trade-off between growth and defense interacts with the selective forces of herbivora and competition to shape locational strategies in plants, Herms and Matson 1992. Thus, a growth differentiation continuum of allocation exists, whereby at one end of the gradient in resource-rich environments, plant competition selects for growth, and at the other end of the continuum in resource-poor habitats, selection favors a differentiation-related strategy of defense. Given that resource limitation affects growth more than does photosynthesis, there should be a curvilinear relationship between allocation to secondary metabolites and resource availability, with peak investment in defensive metabolites occurring at intermediate resource levels, figure 4.14. Thus, in resource-poor environments, both growth and differentiation are limited, but growth is more limited than defense, resulting in slow growth and some investment in secondary metabolites. At intermediate levels of resource availability, growth is limited to some extent, but photosynthesis is less affected, resulting in high investments in secondary metabolites due to excess photosynthesis. Plants growing in resource-rich habitats should not be limited to either photosynthesis or growth, and should allocate a greater proportion of the photosynthetic growth than to defense. There have A. R. E. N. R. G. R. Been few studies examining the growth differentiation balance hypothesis, but 2 studies provide support for the nonlinear relationship between resource availability and allocation to defense. The highest concentrations of terpenes in camphor wheat, Heterotheca subaxillarius (Astracei), occurred at intermediate nitrate levels, Mahaliak and Lincoln 1985, and the highest phenolic content in tomato plants resulted at an intermediate level of nitrogen fertilization, Wilkins et al. 1996. 4.4.5 Synthesis and status of plant defense theory. All 4 of the major hypotheses have made very significant contributions to the theory of plant defense. However, none of the hypotheses is mutually exclusive, and there remains an urgent need for integration, synthesis and further theoretical development, Stamp 2003. The ecologically based growth differentiation balance hypothesis has been expanded to model how herbivora and competition have shaped patterns of allocation to growth, primary metabolism, and defense, secondary metabolism, over evolutionary time, Herms and Matson 1992, figure 4.15. The conceptual model assumes a trade-off between allocation of photosynthesis to growth and defense, with plant competition selecting for enhanced investments in growth and herbivora favoring allocation to defense. Importantly, resource availability mediates the evolutionary outcome. The mix of plant genotypes that survive in the population depends on the pressures exerted by competition and herbivora, with their relative importance being determined by the environment in which the interaction is played out, e.g., resource-rich or resource-poor habitats. Growth-dominated genotypes, genotype A, in resource-rich environments where competition prevails invest heavily in growth and less in defense by evolving phenotypic plasticity, induced resistance, or employing qualitative defenses, figure 4.15a. In contrast, differentiation-dominated genotypes, genotype B, in resource-poor habitats with frequent herbivora invest heavily in constitutive defenses. A stable polymorphism, genotypes A and B, is maintained by disruptive selection in environments in which the evolutionary importance of herbivora relative to competition is high and low, respectively, figure 4.15a. Directional selection exerted on genotypes A and B in environments where herbivora and competition are similarly important results in the evolution of intermediate genotypes, genotype C in figure 4.15b. By subsuming the other hypotheses and making testable predictions, the expanded growth differentiation balance hypothesis is the most mature of the plant defense hypotheses, Stamp 2003. This said, there have been remarkably few studies testing this hypothesis. To further the development of plant defense theory, more experimental tests are clearly needed as is a far more in-depth understanding of the specific biosynthetic pathways involved in the production of defensive metabolites, and the molecular underpinnings of resource-based trade-offs involving plant growth, resistance and tolerance to herbivora. We need to understand much more about how insect herbivores affect plant primary physiological processes, which then cascade to development, growth, yield and fitness effects. An overview of this area is provided by Peterson and Higley, 2001. 4.5 The reciprocal effects of plant-insect interactions on distribution and abundance. Plants vary tremendously in physical structure and they grow in spatially diverse arrangements that range from expansive monocultures to mixed communities comprised of hundreds of species. Moreover, insect herbivores can inflict high levels of defoliation and mortality on their host plants. Thus, it is not at all surprising that plants and insect herbivores can influence each other's distribution and abundance. In the paragraphs that follow, we examine the direct effects of vegetation structure on the density of phytophagous insects and also consider how insect herbivores influence the spatial arrangement of plants. Admittedly, vegetation and habitat structure can indirectly affect insect herbivores by mediating interactions with their natural enemies. Moreover, the spatial configuration of host plant resources and habitat patches, e.g., habitat fragmentation and isolation, can have dramatic effects on metapopulation dynamics, food web dynamics and the diversity of the evolutionary importance of herbivora. Percent of current photosynth allocated to second associated community of arthropods, all topics that we will explore in forthcoming chapters, see chapters 11 to 14. 4.5.1 Consequences of vegetation structure for the distribution and abundance of insect herbivores. Several components of vegetation structure, vegetation texture and architectural complexity, are important contributors to variation in the abundance of insect herbivores, Root 1973, Kariba 1983, Strong et al. 1984a, Dino and Roderick 1991, Kinner et al. 2000, Dino et al. 2005a, b. Vegetation texture has several elements, namely patch size, the area of the plant stand, plant density, the distance between individuals of the same species, and vegetation diversity, the frequency and identity of non-host plant individuals. Also, individual host plants can vary in architectural complexity, which consists of two components, plant size, the spread of plant tissue throw space, and plant part diversity, the number of different plant parts present such as leaves, petioles, stems, buds and fruits. Together, these elements of vegetation structure define the habitats of phytophagous insects, and variation among these structural components can have large effects on herbivore abundance. With his experimental studies on the herbivore fauna of collards, Root 1973 was among the first ecologists to formalize the effects of vegetation structure on the abundance of insect herbivores. He discovered that the flea beetle, Philotreta cruciferae, and larvae of the cabbage butterfly, Pieris rapi, attained higher densities on large, pure stands of collards than on plants growing in isolated rows surrounded by diverse meadow vegetation. Two hypotheses which have guided research in this area of plant-insect interactions emerged from this study. First, the resource concentration hypothesis posits that herbivores, especially dietary specialists, are more likely to find and remain on host plants growing in dense pure stands and thus achieve higher equilibrium densities there. The high frequency of pest problems in agricultural monocultures is certainly consistent with this hypothesis, Altieri and Nichols 2004, Gur et al. 2004. Second, plants growing in mixed vegetation possess an associational resistance to herbivore attack and thus increase lower densities, Tabonanen and Root 1972. The mechanisms underlying associational resistance are many and we will explore these in due course. Before exploring associational resistance, however, we will dissect the components of vegetation texture that contribute to the general pattern of increased herbivore abundance on large, pure patches. Patch size, or the area occupied by the host plant, often has a striking effect on the population density of insect herbivores, Root 1973, Bach 1980, 1981, Ray 1981, Kariba 1983, Strong et al. 1984a, Dino and Roderick 1991, Connor et al. 2000. In general, larger patches of host plants often support a higher density of insect herbivores for several reasons, including increased habitat heterogeneity and resource availability, increased colonization rate and reduced probability for extinction. Indeed, by defaunating islands of the cordgrass Spartina alterniflora in the Gulf of Mexico and monitoring their recolonization by herbivores, it was shown that immigration rates increased and extinction rates declined with island area, wickled to higher densities on large islands. Ray 1981. See also chapters 12 and 14. Importantly, because islands were covered with pure stands of Spartina with similar stem densities, patch area effects were not confounded by either mixed vegetation or plant density effects. Not all herbivores, though, show a positive relationship between density and patch size. Cariba 1983. Certain combinations of life history traits, e.g. high dispersal and concealed overwintering stages, favor colonization and persistence on isolated resources, affect the immigration-extinction dynamic and thus influence patterns of herbivore abundance across a range of patch sizes. Dino 1994b, Hines et al. 2005. For instance, 6 resident species of monophagus planthoppers, delphacity, and leafhoppers, cicadelity, occur abundantly on the cordgrass Spartina patens, which grows as an archipelago of pure patches ranging in size from just a few square meters to immense islands, greater than 20 hectares. These islets are embedded in a sea of another cordgrass species, which is unacceptable as a host for all of the S. patens herbivores. Moreover, these herbivores vary in their dispersal capabilities from flightless to fully flight capable. By virtue of where species spend the winter, either as eggs concealed in vegetation or as active nymphs, they also vary in their exposure to the elements and thus incur very different levels of winter mortality, especially on small exposed patches. The combination of these life history traits explains the relationship between patch size and the abundance of particular sap feeders. The combination of these life history traits explains the relationship between patch size and the abundance of particular sap feeders. Species with low immigration rates, flightless, and high extinction rates, overwinter as exposed nymphs, such as the planthoppers Tumidogena minuta and Apheloma simplex, are least able to maintain populations on small islands, and thus exhibit a positive density patch area relationship, figure 4.16a and b. In contrast, very mobile sap feeders that overwinter as concealed eggs, such as the leafhopper Amplocephalus simplex, persist on and/or effectively colonize small patches and show no significant relationship between density and patch area, figure 4.16c. In this system, the flightless 3000, 1200, 1000, 800, 600, 400, 200, 504 predator Typhus albornatus, hemiptera, which specializes on planthopper eggs, is also rare on small patches and therefore does not likely contribute to the rarity of planthoppers. There figure 4.160. Generalist spiders, however, that emigrate from the surrounding matrix habitat may exacerbate problems of herbivore persistence, especially in small patches. Although other factors certainly play a role, the relationship between colonization and extinction rates is central to elucidating patterns of herbivore abundance in relation to patch area in many systems. Ray 1981, Cariba 1983, Dino 1994b. By planting agricultural crops at different densities in weed-free plots, one can test the effect of plant density on herbivore abundance without the possible confounding effects of mixed vegetation. Dyke et al. 1979, Cariba 1983, Dino 1994b. When this has been done in multiple cropping systems such as oats, soybeans and cucumbers, the densities of the dominant herbivores usually decline with increasing plant density. Another approach to measuring plant density effects has been to assess herbivore abundance in natural settings where plants occur in sparse on dense arrays. Cariba 1983, Marcus et al. 2000. This method has led to highly variable results, with some studies showing a positive relationship between herbivore abundance and plant density, others finding a negative association and yet others showing no relationship. With this approach, plant density is often confounded with plant size, plant nutrition and the diversity of matrix vegetation. In one study where plant size was controlled, the abundance of the parsnip webworm, Depressaria pastinacella, declined with increasing plant density, perhaps in response to altered plant quality. Thompson and Price 1977. Possible mechanisms underlying herbivorous responses to plant density include variation in their colonization-emigration dynamic, altered microclimate and altered interactions with natural enemies. Large plants and structurally complex plants often support higher densities of insect herbivores because they provide a greater variety of feeding and oviposition sites, overwintering sites and refuges from natural enemies than do structurally simple plants. Strong et al. 1984a, Dino 1994b. Just how architectural complexity, both plant part diversity and plant size, can affect the abundance of insect herbivores is revealed in a series of manipulative studies undertaken in the intertidal grasslands of North America. Dino 1977, Ptolemy and Dino 1979, Dino 1980. The grasses Distichylus spicata, Spartina patens and S. alterniflora dominate the vegetation in these habitats, often grow in pure stands, and possess a persistent and deep thatch layer associated with the base of the living vegetation. Each grass is fed upon by a unique assemblage of planthoppers, leafhoppers and plant bugs that sort out into two subcommunities, those that occur above the thatch layer and those that primarily occur within or below the thatch. When the architectural complexity of each grass species was simplified by removing thatch layer, the abundances of sap feeders occupying the different strata changed dramatically. Thatch removal resulted in a large population release for species residing in the upper strata, such as the planthopper Delphicodes detecta ons patens that are normally denied access to feeding and oviposition sites within the thatch. In contrast, lower strata residents such as the planthopper Tumidogena minuta greatly declined in abundance with the loss of the thatch layer, a response that was attributed to the loss of specific feeding and oviposition requirements and not to natural enemies. Plant size in these grass systems also affects the abundance of sap feeders. For example, large plants of Spartina alterniflora support more individuals of the Planthopper Procoliciadalis than small plants, primarily because large plants offer more oviposition and feeding space. Resource abundance, as measured by leaf biomass per plant, also explains a significant amount of the variation in the abundance of insect folivores in other systems such as desert legumes. Marcus et al. 2000. The diversity of the background vegetation in which host plants grow also affects the density of insect herbivores. Root 1973. Atsat and O'Dowd 1976. Khorosheva et al. 2000. Host plants growing adjacent to non-host vegetation often gain associational resistance to herbivore attack and thereby support lower densities of herbivores. Taban and Anand. Root 1972. Root 1973. Kariba 1983. Hamba diaresis CK et al. 2000. Manipulative experiments with corn, beans and squash in Costa Rica showed that the density of monophagous species oafleaf beetles Chrysomelody was invariably higher when crops were grown in monoculture than in polyculture, where the crops were grown in mixtures. Rich 1980, 1981. However, polyphagous beetles obtained higher densities in polycultures than monocultures, emphasizing, as did Root 1973, that the resource concentration hypothesis is best supported by specialist herbivores. The mechanisms underlying associational resistance are varied and involve hindered herbivore searching behavior, changes in host plant suitability, chemical masking of host location cues, or altered interactions with natural enemies, originally stated by Root as the enemies hypothesis. An example of how non-host vegetation interferes with herbivore searching behavior involves specialist leaf beetle Galarucella calmariensis, Chrysomelody, and its host plant Lythrum salicaria, Lythracea, that grows near thickets of the aromatic shrub Myrica gale, Myricaceae, Hamba diaresis CK et al. 2000. When potted Lythrum plants are placed inside and outside thickets of Myrica gale, thus controlling for plant quality, Lythrum plants nested within Myrica thickets incurred lower levels of herbivora and exhibited higher reproductive output in plants outside thickets. Moreover, the pattern of reduced herbivora on experimental Lythrum plants was the same as that observed in natural landscapes when Lythrum grew nearby Myrica. In this study, there was no evidence that the associational resistance was due to increased predation on leaf beetles in Myrica thickets, but rather was attributable to the reduced ability of beetles to find their Lythrum hosts via visual or olfactory interference. Other studies demonstrate that host plant suitability is reduced in the presence of non-host neighbors, thus accounting for associational resistance. Root 1973, Kariba 1983. A unique case involves the hemiparasitic Indian paintbrush, Costylea indivisa Scriphilariaceae, when it is grown in the presence of a lupine host Fabaceae versus in association with alternative grass hosts. Adler 2000, 2003. Costylea growing in close association with lupins sequesters alkaloids from this host and incurs lower levels of herbivora and sets more seed than plants growing in association with grasses or lupine varieties with low concentrations of alkaloids. Associational resistance can also result from higher attack rates on herbivores by natural enemies when their host plants grow in complex habitats surrounded by diverse vegetation. Root 1973, Kariba 1983, Stilling et al. 2003b, Shrewsbury and Raup 2006. Many species of invertebrate predators, for instance, accumulate in complex structured habitat characterized by diverse vegetation, where their negative effects on herbivore populations are enhanced. Dino et al. 2002, Langelato and Dino 2004, Shrewsbury and Raup 2006. A good example of enemy-mediated associational resistance is found in the salt marshes of Florida, where the gallfly Spondylia boraci, Diptera, Societomyidae, attacks two closely related seaside plants, Borachia frutescens and Diva frutescens, Astraceae, Stilling et al. 2003b. In both natural and experimentally manipulated settings, gall densities on Eva are significantly lowered by the presence of Borachia. This occurs because bigger parasitoid species that are abundant in large Borachia galls spill over and attack the smaller Eva galls. Thus, parasitism rates on Eva are higher in the presence of Borachia than where it is absent. The effect is not reciprocal because most parasitoids from Eva galls are too small to successfully attack the large Borachia galls, so gall density on Borachia remains unaffected by the presence of Eva. The small parasitoids with their short ovipositors simply can access fly hosts within large Borachia galls. Not all plants gain associational resistance in vegetationally diverse settings. In fact, the opposite pattern can occur whereby plants incur greater herbivore attack when they grow in mixed species plantings. White and Witham 2000, Rand 2003, Agrawal 2004. Associational susceptibility occurs in two salt-tolerant forbs, Silicornia europaea and Atriplex patula Chenopodiaceae, that grow in the intertidal marshes of New England and are shared by the common leaf beetle, Aeronephala maritima Chrysomelody, Rand 2003. Experimental plantings of Atriplex with and without Silicornia as a neighbor showed that the presence of Silicornia enhanced beetle attack on Atriplex and lowered its survival and reproductive output. Moreover, surveys of natural marsh habitats found that Atriplex growing in close association with Silicornia was attacked more by the chenopod specialist than plants not neighbored by Silicornia. Also, polyphagous herbivores that reach outbreak proportions often move to nearby less preferred host plants, a spillover effect that results in associational susceptibility. White and Witham 2000. For example, when larvae of the fall cankerworm, Alsophylla pomateria, reach high densities on their preferred box elder, Acer negundo, host, they disperse to defoliate adjacent cottonwood trees that when growing in open settings incur little herbivora from this moth. Associational susceptibility results from a different mechanism, namely the proximity of oviposition sites, in a milkweed grass system. Agrawal 2004. The milkweed beetle, Tetraopes tetrophthalmus coleoptera, Cerambicity, feeds on the milkweed Asclepias syriaca in old field habitats in southern Canada. In addition to its developmental host, beetles also require grass stems for oviposition, even though they don't consume them. As a consequence, milkweeds growing in association with grasses incur significantly higher levels of herbivora and reproductive losses from adult beetles than do milkweeds growing alone. Moreover, milkweeds suffer root competition from grasses, an effect that is exacerbated by beetle herbivora. In contrast, grasses enjoy competitive release by facilitating